welcome to today's webinar. My name is Melissa and I'm a Scientific Training Officer in the training team at EMBL-EBI. I will also be your host for today's webinar. So in today's webinar, we have Mihai with us and he's going to be talking to us about PDBe-KB Aggregated Views and presenting PDB data in a different context. So with that, I'm going to hand over to Mihai and we can get started with the webinar.
Welcome everyone to this webinar. I'm Mihai Váradi from the PDBe team, the Protein Data Bank in Europe. And today I will be talking about a new type of data access and visualization method that we are developing here at PDBe. First, I will give an introduction to PDBe-KB, which is the Protein Data Bank in Europe knowledge base. And in order to understand what motivates PDBe-KB, the development of PDBe-KB, I would like to start with a simple example. If you look at this structure, it's not giving you too much information. You can see some of the secondary structure elements, but you can't really say what is it. And even if you look at the structural context, you see that it's part of a complex, yes, but what does it do? So the point I'm trying to make is that the coordinates by themselves, they just specify the shape and may not be of too much intrinsic biological value unless they can be related to other information. So the idea behind the knowledge base is that we take molecular data and we translate it, compile it, and present it in such a way that you can use it as information to derive knowledge and to gain a better understanding of a certain biological system. So that ideally you will be able to say, why is this mutation having an effect and causing a certain disease, not just being able to see how the structure looks like. So PDBe-KB is trying to place the structural data in its biological context by establishing a community-driven and integrated resource. This is basically a collection of structural annotations, and the idea is that this will promote both fundamental and translational research. I will try to briefly describe how this works in practice. And on the left-hand side, you can see that traditionally, the protein data bank is focused on single PDB entries. For example, in this case, you can see that there are 3 different PDB identifiers for 3 different structures. And in fact, these are domains from the same protein, but covering different sections of the protein. And the problem is that the way PDB currently works is it's not trivial to gain this information. It's actually part of the same protein and where these are located. Also, these structures are used by many specialist databases and software developers who take these structures and derive information from them. So they put their own annotations on top of these structures. For example, this might be ligand binding sites or sequence conservation scores, or maybe an interaction interface annotation, and so on. The idea behind the knowledge base is that all these specialist databases and software developers, they provide these annotations in a unified manner. And then here at the KB, we integrate all these and connect all these annotations to the structural data that we already have in PDB. And the reason why this is possible is because there are more than 200 specialist resources who use PDB data and put their own annotations on top of these structures. And like mentioned before, today I will be mostly focusing on a new way of accessing this data and visualizing the structural data in a different context. The way you can reach these protein-centric pages are described here. On one hand, you can use a URL like this, where at the end you either have a PDB identifier or a UniProt identifier. You will also be able to get here from the current PDBe entry pages, and also there is a sort of landing page at the PDBe-KB URL found here. So now I will walk you through the different segments and functionalities that we have on this protein-centric page. These pages start with a summary section, which gives you an overview of the information that we have, the structural information that we have for a certain protein. For example, in this case, what you can see is we have a box which gives you textual summary of this protein. You see the name of the protein, you see the gene name, and also flags for enzymes. And in fact, these are links to Brenda, so you can learn more about the specific enzymatic activity that this protein performs. And also, if this protein is known to be implicated in disease, then we have detailed information on that too. We also have description of the biological function, and this actually is coming from UniProt. Then on the right-hand side, we try to give you an overview of all the available PDB structures for this full-length protein. So I will go into a bit more details regarding this component. At the top of this visualization component, you have a help text tooltip. So if you were to go there, you would see a brief description of what we have available within this component and what is it that you see here. Underneath, at the bottom of this component, you can see a link which goes directly to the chain that is being displayed here. And under that, you get information regarding the coverage, which basically means that this particular chain, for which you see the structure here, is found at these residue ranges in terms of the UniProt sequence. And this actually is 10% of the full-length sequence of UniProt. There are several ways, or a couple of ways, to navigate between the different PDB entries mapped to this UniProt sequence. One is to use these arrows, and the other is to use these boxes. Each of these boxes represent a specific PDB chain. The currently selected one is always blue, and if you click on another one, then it will turn to that structure and show it there as well. If you click on the image itself, then it will link to a 3D visualization tool, Lightmole. And this is an interactive tool where you can get into the details of the structure itself. Finally, at the summary section, we have ways to navigate between the different parts of the page. And at the top, we have a navigation bar. And also we have these icons, which on one hand serve as navigation. So these are buttons that you can click on. And also they give you an overview of the available data. So for example, in this case, what you can see is that for the crab binding protein, we have 88 structures, 81 unique ligands observed in PDB. This protein is found in 5 interactions with other proteins or nucleic acids. We have 3 additional functional annotations, which I will detail later. And we have almost 400 publications. So this covers the summary section of the page. We are using a sequence feature viewer called ProtVista, and now I will describe the functionalities that this tool has. ProtVista is developed at EBI in a collaboration between UniProt, PDB, and Interpro. And the idea is that there will be this one common tool used by all these different resources so that there is a unified way of visualizing data. We, on this page, use ProtVista at multiple locations, as you will see. At the top of ProtVista, you have a scale, which goes from 0, actually from 1 to n, and this is representing the sequence, the UniProt sequence of the protein. There are so-called main sections. So for example, in this case, we have the PDB structure section, domains, and secondary structure. And underneath, we have tracks, which have the actual data for each of these sections. These main sections can be collapsed, and then you can get an overview of the available data. So all the tracks are sort of summarized here. There are 3 main buttons that you can use. The first is resetting the whole visualizing tool. The second is for zooming in, and the third is for hiding specific tracks. So first, the reset button. In case you zoom in at any of these regions or annotations, or if you remove or hide tracks, you can just press this button and then it will reset the view to its default. Next, we have a button for zooming. And in fact, there are more than one ways of zooming. Probably the most convenient is to use the mouse scroll bar or scroll button, and that will zoom into the particular section. But you can also use this button, and there you can specify the to and from region where you would like to zoom in, either only just highlighting that region or zooming onto that region. If you zoom in sufficiently strongly or highly, you will be able to see the amino acid sequence itself. Next, if you press the hide button found here, then you can choose to hide certain main tracks, main categories. So for example, in this case, we can hide the domains and the secondary structure and only see the coverage. And again, if you wish to reset, then you can use the reset button. If you hover over any of these colored segments, colored boxes, then you will see a tooltip which gives you more detailed information on what exactly is this region about. And finally, if you look at the subtracks, there are two additional buttons. The first is, if you click on this, it will zoom on this track. And then the second, if you click on that, it will remove or hide this track completely. And again, using the reset button can reset the view to default. Like I mentioned, we are using ProtVista in several locations, and one such location is the structural coverage part. In the structures and domains section, we use ProtVista to show the coverage of the UniProt sequence in terms of the available PDB structures. These are the PDB entries that are available for this particular sequence, and these are sorted first by the number of observed residues and second by resolution. In each row or each track, you see the link to the PDB entry, and also you see these icons which link to certain parts of the PDB entry pages. So for example, the first will get you to the experimental details, and then the second will give you information about is it found in a complex or not. If it's grayed out, then it's not in a complex. If it's green, then it's interacting with a macromolecule. Next, if this icon is green, this means that it's interacting with nucleic acid. If it's gray, then it's not. Then this icon signifies if the protein is interacting with small molecules, ligands, or if it's not. And if you click on it, it will go to the ligand-specific page of PDBe. And finally, we have a link to the publication where this entry was described. And we also give you information on the resolution of this particular entry. Under the coverage section, we have information on the domains mapped on top of the UniProt sequence. We have PFAM, CATH, and SCOPE domains mapped. And finally, we have a section for secondary structures. And these secondary structure elements are actually from the representative PDB chain for each covered region. So for example, in this region, we chose the best representative PDB chain and derived a secondary structure element from that. And finally, if you would like to download the data that is available in the ProtVista viewer, then you can choose to download in either CSV or JSON format. On the protein-centric pages, we have a section dedicated for ligand binding sites and ligands that are observed to be interacting with the protein of interest. This section is made of two main parts. At the top, we have a gallery, and underneath that, again, we have a ProtVista sequence feature viewer. At the top, you can see two numbers. One is the number of directly interacting ligands, unique ligands or distinct ligands, and the other is all the available ligands. The difference is that on one hand, you have all those ligands which are directly interacting with the protein of interest, meaning that in the PDB entry, you see that this chain is interacting directly with these molecules. On the other hand, you see all the ligands which are observed in the same PDB entry as the protein of interest. There is a search field which you can use to filter the gallery, this being the gallery, and you can use either the molecule name, the unique molecule code, which is we refer to as head code, or the PDB identifier. Using any of these, we filter the gallery underneath. So for example, starting to type in sulfate, it will filter down the gallery to that specific ligand. By default, we are showing only those ligands which are directly interacting with the protein of interest, but you can tick this box and then you will be able to see all of them. So by default, we only show these 18 directly interacting ligands. Clicking this will show you all the 81 ligands. If you mouse over or tap on any of these unique identifiers for the ligands, then you will be able to see the full name of that ligand. Under that, you can see a link, and in fact, this link goes to the PDB search, and it will list all the relevant PDB entries where you can see this ligand together with the protein of interest. And finally, if you click on the small image of the ligand, then you will get to the 3D visualization. This will be by default focused on the first instance of this ligand occurring in contact with the protein of interest. It can happen that you have this ligand in more than one PDB entry, and in this case you will see how many entries is this ligand observed in. And in that case, if you go to the 3D visualization, you can use this drop-down to choose which PDB entry you would like to see. Under the gallery section, we have the sequence feature viewer. And here we are showing all those residues of the protein sequence which are directly interacting with a number of ligands. If you click on any of these highlighted segments, then you will see more information regarding which residues are there, the range of them, also which ligand are they binding to, and an example entry where you can go and see the details. If you collapse this track for the ligand binding sites, then you get an overview of where all these different binding sites are located, and you can discover the pattern of those residues which are consistently binding to small molecules or ligands. And finally, if you would like to download this data that's being displayed in the sequence feature viewer, then you can choose between JSON and CSV formats. There is a dedicated section for interaction interfaces and interaction partners on this protein-centric page. At the top, we have a gallery, and underneath the gallery, we have a sequence feature viewer implemented. At the top part in the gallery, you can filter what's being shown using either the protein name or the UniProt accession or the PDB identifier. If you mouse over or tap on any of the UniProt identifiers that you can see here, you will be able to see the full name of the molecule. Under that, you have a link which goes to the PDB search, and it will list all the entries where you can see the protein of interest and this particular interaction partner. And under that, there is a button, and clicking on that will go to the protein-centric page of that particular protein. So it will basically show you the other side of this interaction and all the details. So a similar page like this, but for that other protein. Finally, if you click on the small image, then you will be able to display it in 3D. If there are more than one entries associated with this interaction, then you will see this by the number, and if you display it, you will be able to choose which PDB you would like to visualize using this drop-down at the top. Under the gallery, we have the sequence feature viewer, where we are showing all those residues of the protein sequence which are observed to be interacting with its partner. So these are basically the interaction interfaces. In case of homomeric interfaces, we use a different shade of blue so that immediately you will see that this is a homomeric interface as compared to the other types of interfaces. If you mouse over or click on any of these segments, you will get additional information, in particular the range of the residues involved in this interface and also the partner, and the link to one of the PDB entries where you can actually see this interaction. If you collapse the track, then it will give you an overview of all the interaction interfaces, and you can see where you have promiscuous interfaces, residues that bind or interact with more than one partners. And if you would like to download the data that's displayed in the sequence feature viewer, you can choose between CSV and JSON formats. There is a section focusing on additional annotations, functional annotations, that are derived from the structural data. Here we have either predicted or evidence-based annotations coming from PDB partner resources. In case of predicted annotations, they are colored gray so that it's immediately distinct visually that these are different than the usual annotations. These are predictions. In case of these annotations, you have two new type of links. The first on the left-hand side is going to the page of the particular resource, the PDB partner resource. And here you can learn more about what exactly the data is and how do they calculate it. While on the right-hand side, the source link should ideally show you the detailed annotations for a particular entry. So not just describing the method and how this was calculated, but the particular annotations, the complete data set of annotations for this particular entry. Under these additional annotations, we repeat the sections for domains, ligand binding sites, and interaction interfaces. And the reason for this is that it allows the users to compare these different types of annotations. Finally, if you are interested in downloading this data, then you can choose either JSON or CSV formats and download that. The bottom section of the protein views is dedicated to publications. And here we have three main categories of publications collected for this particular protein. The first is a collection of PDB publications. This basically means that these are the publications where the PDB entries have been described originally. Next, we have a section dedicated to reviews where the PDB entries have been mentioned. And finally, we have a section for publications from UniProt that correspond to this particular protein. You can navigate between the different pages of these sections by either using the arrows or the numbered buttons. Inside the table, you have links to EuroPMC, or you can use the DOI as a link as well to get to the publication. And when available, we provide direct links to the PDB entries which are related to this particular publication. So these were the various sections of this aggregated view, and this was the introduction to the PDBe-KB as a resource. And now I would like to open the chat box for questions.
Okay, thanks very much, Mihai. That's really interesting to see how that PDBe-KB is coming together. We do have time now for questions. So I know there's some coming through in the chat box already, but while you do add your questions to the chat box, just a reminder that this is part of our regular Wednesday webinar series. So we have a whole range of webinars that we run every second week on a Wednesday afternoon UK time on various different topics. You can find the full list of upcoming webinars on our website, www.ebi.ac.uk/training/webinars. Okay, so the first question that we have is what happens in graphical features of Sequence Viewer when downloaded as a CSV? For example, residue ranges, colored blocks of interacting residues. Are these converted to numerical ranges?
Yes. So the downloaded CSV is basically a table with text labels and the numeric values of the ranges. But if you choose the JSON format, it also includes the colors as well, if you, for some reason, want to retain that information as well.
So the next question is, with regards to the functional data, does it provide confidence scores to functional annotations for prioritization?
Yes, that's a very good question indeed. The way it works on this particular type of visualization is that we give an aggregated view on the annotations available. And our next step now is to actually link this to specific entry pages where you will get all this information. So we have, in most of the cases of these resources, they provide a row score that they calculated, a confidence score that they calculate, and also a classification of the confidence, which is basically low, medium to high. The reason being that the number they use for confidence score can be very different between different resources. But yes, that data will be certainly available.
So there's a question about whether these entries provide links or information across species for a particular protein. Is that something that you can do?
It's something that you at the moment cannot do, but we are actually working on this actively. The reason why it's not there yet is because we are not clear how to actually implement this in the best possible way, but this is something that we certainly want to add in the coming months.
Okay, thanks for that answer. The next question is about Snips. Do you have plans to enable annotation of Snips onto proteins?
Yes, absolutely. The first step will be to take all the available information from UniProt. And what we are working on at the moment is that these types of annotations, they need a special type of track. So the one that we are currently using with these boxes is just not suitable for it. And we would like to take what UniProt is using currently. But this ProtVista version that you've seen on these pages, it's a new version that's actually not have been released yet. And so this particular track for variation and mutation data is still being created now, but this certainly will be there, yes.
Okay, thank you. And how frequently will the database links be updated and synchronized?
Every week. The plan is to have this updated every Wednesday.
And related to that, when is PDBe-KB going live? Live?
It's going into production next Wednesday, so that will be the official release date.
Okay.
The URLs are already working, so you can already play around with this, but the official release will be next Wednesday.
Okay, so I guess we should keep an eye on the Twitter channels and things like that for the big announcement. One last question coming in just now is, will there be a possibility to carry out homologue identification through the interface?
Yes, that's certainly something that we are looking into. Yes, absolutely.
Okay. All right, so we're going to leave it there for today. If you do have any more questions, I'm sure that Mihai and the PDBe team will be happy to answer them. So get in touch with the help desk if you have any of those. Otherwise, thank you very much, Mihai, for the webinar today.
No problem.
And thank you all for joining us. We hope to see you in another webinar again soon. Thank you. Bye.